Imagine holding a miniature version of a human organ right in your hand. A tiny living structure that breathes, maybe beats, or even thinks,
Mhm.
all grown in a lab.
Yeah, it sounds like science fiction, doesn't it?
It really does. Today, we're diving deep into this incredible world of organoids.
And look, this isn't just some futuristic concept anymore. It's a field that's advancing, well, incredibly rapidly. It's genuinely revolutionizing medicine as we speak.
Right. So our mission today, really, is to give you a shortcut to getting up to speed on organoids. We'll unpack what these mini organs actually are, trace back their, well, surprisingly long history,
Mm-mm.
explore how scientists actually create them, and reveal some of their groundbreaking applications. Stuff like personalized drug testing,
Mm.
even the potential for organ transplantation.
It's huge, but we also need to look at the hurdles, right? The challenges they still face and where the tech is likely headed next.
Exactly. Get ready for some serious aha moments, folks.
Yeah.
This stuff could really reshape how you think about biology.
So let's start with the basics for this deep dive. What exactly are organoids?
Good question. Lay it out for us.
Okay. So at their core, they're three-dimensional cultures,
Mm.
3D cultures, usually derived from stem cells.
Okay, stem cells.
Right. And you put these cells in an environment that sort of mimics the body's natural scaffolding, the extracellular matrix or ECM.
Yeah.
And here's the amazing part. They self-organize. They arrange themselves into structures that actually resemble miniature organs.
Self-organized. Oh, that just sounds wild.
Mhm.
Can you break that down a bit? It sounds almost automatic.
It is, in a way. I mean, you give them the basic building blocks, the right sort of environmental cues, and these cells, they kind of instinctively know how to assemble themselves. It's not like scientists are giving them a step-by-step blueprint.
Wow.
Yeah. And they contain multiple cell types, just like the real organ they're mimicking, and they can actually recapitulate, perform some of that organ's specific functions.
So like what? What kind of functions?
Well, think about it. A heart organoid might actually contract, beat. A brain organoid can show neural activity. A little liver organoid might do some basic filtration.
That's incredible.
It is. Their cells grouped together, they're spatially organized. It really resembles a tiny living organ piece.
Okay, so how tiny are we talking?
Huh.
Microscopic. Or can you actually see these things?
They can range quite a bit. From smaller than the width of a human hair, maybe up to about 5 millimeters.
5 millimeters, so like a tiny sugar cube almost.
Sort of, yeah, for some. And look, for this whole organoid culture thing to really work, the cells have to be able to do that self-assembly into these organized tissues, mimicking how organs develop naturally.
Right.
Plus, the stem cells inside, they need to keep their ability to self-renew. That lets scientists grow them over long periods, which is, well, absolutely key for research.
You know, it sounds so cutting edge, like something we just figured out. But what's fascinating is that this idea, it's not entirely new, is it? The history goes back further than you'd think.
Oh, way back. Actually, 1907.
1907.
Really? Yeah, Henry Van Peters Wilson. He did this pioneering work showing that if you separated sponge cells, they could spontaneously reorganize themselves and basically reform the sponge structure.
From sponge cells, wow.
Exactly. It was this really early, kind of astonishing hint that these basic biological building blocks have this inherent ability to self-organize, a principle we're only now really harnessing properly.
So even over a century ago, this idea that cells could just reassemble, that was being observed.
Mm-hmm. Precisely. It laid a crucial bit of groundwork. Then, fast forward to the mid-20th century, maybe the 50s and 60s, labs started using cell isolation organ regeneration techniques more actively.
Okay.
The first mammalian organoids were actually developed over 60 years ago.
Yeah.
And there were even early experiments trying to grow things like kidney, skin, liver structures from chick embryo cells.
Right, but the modern era really kicked off with stem cells, didn't it?
Right.
Pluripotent stem cells, PSCs. What was it about them that just turbocharged the whole field?
Well, it was the ability to finally work with and control these incredibly versatile stem cells.
Yeah.
Understanding their spontaneous regeneration, it just changed the game entirely.
How so?
Scientists could suddenly think beyond just observing. They could actively explore development, maybe even think about repairing damaged organs. You had PSCs first from mouse embryos, then human blastocysts.
And then iPSCs, right. Induced pluripotent stem cells.
Exactly. That was another massive leap, being able to create these versatile stem cells from, say, just a skin sample. Huge.
And I know Howard Green made a big contribution around 40 years ago, too.
Yes, absolutely. Howard Green's work was groundbreaking. He managed to propagate human epidermal keratinocyte skin cells using these special mouse embryonic fibroblasts, and they could actually mimic human skin in vitro in the dish.
A functional skin model, basically.
Yeah, a huge step towards more complex, functional tissue models.
So it really feels like the 21st century is where organoids properly took off.
Yeah.
They started being seen as maybe a better alternative to animal models, right? Especially as the stem cell tech got better.
Definitely. And their applications just started expanding rapidly toxicology testing, thinking about organ transplantation, cancer research.
Yeah.
And, there's a really memorable detail that highlights why 3D cultures are just so much better than the old flat 2D ones.
Oh, yeah. What's that?
Researchers found that breast epithelial cells, when you grow them in 3D, could form these duct-like structures, and they could actually synthesize milk proteins.
No way.
Yeah, something you just don't see in flat 2D cultures. It really drives home how that 3D environment lets cells keep their proper characteristics, behave more naturally.
Like they need that context to remember who they are.
Exactly. It's about those complex interactions between cells.
Which brings us perfectly to the next point,
Mhm.
the organoid advantage. It really sounds like they're a massive upgrade over the older models. So let's break that down. Why are they so much better? What were the problems with, say, traditional 2D cell cultures?
Well, okay, 2D cultures. They're cheap, they're relatively easy to work with, sure, but they fundamentally lack that true three-dimensional architecture you find in real organs.
They're flat.
They're flat, so the cells, their morphology, their shape gets altered. They often lose important markers on their surface. And critically, they just can't accurately predict clinical outcomes or mimic those really complex tissue environments.
Right.
You can't simulate the microenvironment properly. It's like trying to understand how a city works just by looking at a flat map. You miss all the depth, all the interactions.
Good analogy. And then animal models. We've used them for ages because, well, physiologically they seem similar.
Yeah.
But they have big drawbacks too, right? I mean, how often do animal results just
Yeah.
not translate to human?
Oh, very often. That's a huge challenge. Species differences are significant. A classic example is Yersinia infection. Pigs can carry it without symptoms, but humans get severe diarrhea. And there are tons of human-specific mechanisms that you just can't model accurately in animals. Plus, animal models. They're expensive, need specialized facilities and staff, and of course, there are major ethical considerations for many people.
Okay. So organoids step in as this revolution, you called it. What are their key strengths? Why are they bridging this gap?
They provide a 3D microphysiological platform. That's the key. It closely mimics human tissues and organs, allowing for much deeper, more relevant research.
That's just human-like.
Exactly. Which means they're not just a small step forward. They could fundamentally shift how we discover drugs, potentially saving billions on failed clinical trials, speeding up breakthroughs.
That's a big deal.
It is. They bridge that critical gap between the lab bench, the preclinical studies, and actual clinical applications. They offer, generally speaking, high genetic stability, excellent capacity for expansion and differentiation,
Right.
and that remarkable self-organization we talked about.
Mhm.
So cells function much more like they would inside the body. And crucially, they contain multiple cell types, replicating those complex cell-to-cell and cell-to-matrix interactions that are just vital for how an organ actually works.
They capture the complexity better.
Precisely. They keep the benefits of older models, but bypass a lot of the main limitations.
Okay, so we know what they are, why they're potentially better,
Mhm.
but the really mind-bending part is how scientists actually make them. What are the ingredients, the raw materials, and what kind of environment do they need to coax these things into existence?
Right, the building blocks. Well, organoids can be grown from various cell sources. You've got pluripotent cells, which are super versatile.
Like the ESCs and iPSCs we mentioned.
Exactly. Or tissue-resident stem cells, sometimes called adult stem cells. Progenitor cells, which are a bit more committed or even fully differentiated somatic cells, regular body cells.
Okay, so let's talk about those stem cells. Adult stem cells, or ASCs, where do they come from and what are they good for?
ASCs are often used for tissue-specific organoids. Like intestinal organoids are often grown from stem cells found in the crypts of the small intestine or colon. And these tend to maintain genetic stability and the unique characteristics of that specific organ. They're used for liver, kidney, lung, prostate, stomach organoids too. Certain signaling pathways, like Wnt, are really crucial for keeping them growing long-term.
One signaling, like a molecular switch.
Kind of, yeah. It guides growth and cell identity, making sure that the mini organ develops correctly. It's essential for many of these ASE-derived organoids.
Then there are the induced pluripotent stem cells, iPSCs.
Yes.
You mentioned taking skin cells and reprogramming them. That still sounds like magic. How does that work?
Haha, well, it's a complex biochemistry, but essentially scientists use specific factors to reset these adult cells, turn back their developmental clock to an embryonic-like state.
So they become pluripotent again, able to become anything.
Pretty much. That versatility makes them ideal for creating models where maybe adult stem cells are hard to get or don't work as well. Things like kidneys, brains, retinas.
Wow.
And you can do cool things like with iPSC-derived intestinal organoids. If you introduce neural crest cells involved in nerve development, they can actually start to mimic gut contraction. You get a little nervous system in there.
That's amazing. And finally, embryonic stem cells, ESCs, the sort of original pluripotent stem cell.
Right. Derived from blastocysts. These are highly plastic, very versatile. They can form incredibly complex structures, things mimicking the cerebellum, the optic cup in the eye, even hippocampal-like structures.
So they're good for early development models.
Exactly. ESCs let researchers model those very early embryonic or fetal stages of organ development, which is invaluable for understanding birth defects, congenital diseases.
Okay, so you've got your cells, but the environment is critical, you said. The culture medium, the scaffolding. What goes into that?
The extracellular matrix, the ECM scaffold, is absolutely fundamental. Think of it like the structural support and signaling hub for cells in our body.
Right.
Traditionally, something called matrigel has been used a lot. It's an ECM extract derived from mouse tumor cells, and it works pretty well.
But mouse-derived. I sense a but coming if you want to use these clinically eventually.
You got it. Its xenogenic origin coming from another species and the fact that it can vary quite a bit from batch to batch, that makes it less ideal for clinical use and can affect research reproducibility.
So what's the alternative?
Synthetic hydrogels are really promising. These are lab-made materials. The big advantage is they have a precisely defined composition.
Meaning more consistent results.
Exactly. And they're tunable. Scientists can control things like stiffness, pore size, how quickly they break down. They can even embed specific bioactive molecules right into the gel.
Tailor-made environments.
Precisely. Polyisocyanide, or PIC gels, are one example. You can really customize the microenvironment for the specific organoid you're trying to grow.
That level of control sounds amazing. What about physical forces? Like, does the stiffness of the gel actually matter?
Oh, hugely. Biophysical factors are very influential. The elasticity, the stiffness of the culture material can literally guide stem cell differentiation.
How so?
Well, generally, softer materials might encourage cells to become neural types, while stiffer materials can promote bone or muscle development. There's been work with pancreatic organoids cultured on soft hydrogels, specifically to enhance the differentiation of beta cells, the ones that make insulin. Really important for diabetes research.
Fascinating.
Yeah.
And oxygen levels. Seems basic, but crucial, right?
Absolutely vital. You need the right oxygen concentration. Sometimes that even means hypoxic environments, low oxygen, which is actually critical for developing certain types like brain and tumor organoids. It helps mimic conditions inside the body and study things like drug resistance.
So it's not just the physical structure, it's also chemical signals, growth factors, other molecules floating around telling the cells what to do.
Precisely. Soluble signals are crucial guides. Growth factors like EGF, epidermal growth factor, or FGF, fibroblast growth factor, are often added to promote growth and differentiation in many types of organoids.
And you mentioned signaling pathways before, like WANT. Are there others that are key?
Yes, several. WANT is critical for many, like intestinal, liver, pancreatic organoids, but you have to control it carefully. Too much can lead to uncontrolled growth, like tumors.
Right.
Notch signaling is another big one. It heavily influences what fate stem cells choose like, whether they stay a stem cell or become a specific type, and helps maintain the overall tissue balance within the organoid. Think of it as managing the cell career paths.
It sounds like this incredibly delicate molecular choreography.
It really is. And small molecules play a huge role too. These are often used to fine-tune the process. They can push cells towards a specific type or modulate those signaling pathways with more precision. For instance, specific small molecules are used to activate certain channels in cystic fibrosis organoids for drug testing or to help model tumor development. They're like precision tools for guiding the whole process.
Okay, beyond these core methods, are there even more advanced techniques pushing the boundaries, making organoids more complex or functional?
Oh, yeah, definitely. One important one is air-liquid interface culture, or ALI.
Air-liquid interface.
Yeah, basically you grow the cells at the boundary between the culture medium and the air. This gives them much better oxygen supply and really promotes differentiation, especially for things like lung organoids where that air exposure is natural.
Makes sense for lungs.
Mm-hmm. It helps them mature better, develop more realistic features.
And I've read about organoid on a chip systems. That sounds very futuristic. What are those?
Right, that's microfluidic-based culture. It's a really advanced approach. You create these tiny microscale environments on a chip.
Like a miniature lab.
Sort of. It allows incredibly precise control over things like nutrient gradients, waste removal, even fluid flow mimicking blood flow.
And what's the benefit?
This leads to more mature, more functional organoids. They often develop better structures, sometimes even rudimentary vascular networks, tiny blood vessel beginnings, and it enables really detailed disease modeling.
Can you give an example?
Sure. In kidney organoids, these on-a-chip systems helped researchers identify signaling pathways involved in polycystic kidney disease that just weren't apparent in simpler static cultures. It's about creating a more dynamic, realistic microecosystem for the organoid.
Okay, this is all incredibly sophisticated, which brings us to the big question. What can we actually do with these things? What are the applications where organoids are really starting to unlock potential?
Well, one huge area is disease modeling. Organoids give us these fantastic microenvironments to study how human tissues interact with pathogens, viruses, bacteria, parasites.
Like with COVID-19, SARS-CoV-2.
Exactly. Lung organoids were, and still are, critical for studying SARS-CoV-2 infection, how it affects cells, how it replicates, and for testing potential treatments directly on human-like lung tissue.
Wow.
And intestinal organoids. They can even reconstruct the whole process of how a virus invades the gut, helping identify genes that make us susceptible. It's an unprecedented window into infection.
What about non-infectious diseases, like genetic conditions?
Yeah, huge potential there too. You can derive organoids directly from patients with genetic diseases, or you can use gene editing like CRISPR to introduce specific mutations into healthy organoids.
So you can build a model of the disease.
Precisely. This helps researchers understand exactly how the disease mechanism works at a cellular level. And crucially, it provides a platform to screen for drugs or therapies that might correct the defect.
Like for cystic fibrosis.
That's a prime example. Intestinal organoids from CF patients have been used extensively to test drugs that could restore function to the faulty protein. For visual disorders, too, like retinitis pigmentosa, gene-edited retinal organoids are helping understand the defects and test potential cures.
And cancer. You mentioned tumoroids before. How are they being used?
Tumoroids are incredibly valuable. They're essentially organoids grown directly from a patient's cancer cells.
So a living biobank of someone's tumor.
Exactly. They let scientists study the specific mutations in that tumor, understand how genes lead to cancer traits, figure out the molecular basis of how that tumor grows and develops. They are becoming invaluable preclinical models.
Preclinical, meaning before human trials.
Right. Especially for personalized drug screening. There was a study on pediatric kidney cancers, for example, where they grew tumoroids from different cancer subtypes. They could analyze their features and test how well standard chemotherapy drugs actually worked on each specific tumoroid.
So the idea is maybe one day take a biopsy, grow the tumoroid, test drugs on it first.
That's the vision. Test drugs on your specific cancer outside your body to find the best treatment for you before you even start therapy. Personalized oncology.
That naturally leads us into drug discovery and personalized medicine more broadly. This sounds like it could totally change the game for pharma companies and patients.
Oh, absolutely. Organoids are really poised to revolutionize drug development. Think about the huge failure rate of drugs in clinical trials.
It's massive, right? Something like 90% fail.
Easily. And the cost is astronomical, averages over $2 billion to bring one new drug to market. A lot of that failure stems from poor preclinical models, 2D cells, animal models that just don't predict human responses well.
So organoids could help predict toxicity or effectiveness better.
That's the hope. They can potentially predict toxic effects and effective doses more accurately. You can evaluate combination therapies, and using patient-specific organoids, you might even forecast clinical outcomes better.
Patient-specific.
Yeah.
Using those iPSCs again.
Exactly. Imagine creating biobanks of iPSCs derived from diverse patient populations linked with their clinical and genomic data. You could devise truly individualized treatment plans, even for rare diseases where you only have a few patients worldwide. That potential for a limitless supply of patient-specific tissue is huge.
Okay, let's shift gears slightly. Regenerative medicine and organ transplantation. You called organoids a hopeful remedy. That's a strong phrase.
I think it's justified. Organ transplantation faces massive hurdles, donor shortages, the need for lifelong immunosuppression drugs with nasty side effects.
Right.
Organoids offer a potential way around that. The idea is to use them to replace damaged tissues or even whole organs, perhaps combined with gene editing to avoid rejection or nanotechnology for better integration.
Has this actually been done, moving from lab to animal?
There's progress. For example, researchers grew colon epithelium organoids from stem cells and transplanted them into mice that had colitis, like inflammatory bowel disease. And they found the organoids successfully engrafted and helped the damaged tissue recover.
Wow. So it's not just about studying disease, it's potentially about growing the cure, the replacement parts.
That's a long-term vision, yes. A very exciting one.
Now, something else fascinating you mentioned earlier, extracellular vesicles, EVs. What are they and how do organoids help us study them?
Right, EVs. Think of them as tiny little packages or vesicles released by cells. They're wrapped in a lipid membrane and contain molecules like proteins and RNA.
And they travel between cells.
Exactly. They act as messengers, mediating cell-to-cell communication, and they're increasingly recognized as important biomarkers for diseases, especially cancer.
So how do tumoroids fit in?
Tumoroids are excellent 3D models for studying how cancer cells produce EVs and what role those EVs play in cancer progression, things like promoting tumor growth, oncogenesis, helping cancer spread, metastasis, or even manipulating the immune system.
So we can use mini tumors to eavesdrop on cancer cell communication.
Pretty much. And the insights can be deep. For instance, one study found that EVs derived from cervical cancer tumoroids were very similar to the EVs circulating in the blood of actual patients.
Meaning they could be used for non-invasive monitoring, like a liquid biopsy.
That's the suggestion, yeah. But it gets even more interesting. EVs can potentially be engineered.
Engineered how? As treatments.
Yes, potentially as nanotherapeutic agents. You could load them with specific molecules like drugs or therapeutic RNA designed to target and inhibit cancer growth. For example, researchers have loaded EVs with a specific microRNA, miRNA 497, which seemed to inhibit non-small cell lung cancer growth in models, possibly by cutting off the tumor's blood supply.
Targeted drug delivery using the body's own messaging system.
That's the idea. Huge potential there.
Okay, let's zoom in now on some specific types of organoids. What are the stars of the field right now? Let's start with brain organoids, the mini brains.
Right, brain organoids. These are amazing. They can mimic aspects of human brain development quite closely, forming complex structures with different types of neurons and support cells. They can even form functional synaptic networks.
So they're making connections, like real neurons.
Yes, to some extent. This makes them incredibly valuable for studying complex neurological disorders. Think Alzheimer's, Parkinson's, autism spectrum disorders, where animal models often fall short, and of course for testing potential new drugs for these conditions.
Okay, next up, intestinal organoids.
These model both the small and large intestine. They're workhorses for understanding gut biology and disease, inflammatory bowel disease, IBD, cystic fibrosis, infectious diseases. They're great for drug screening, obviously, but also for studying the really complex interplay between our gut lining, our immune cells, and the trillions of microbes living in our gut, the microbiome.
And lung organoids. We touched on SARS-CoV-2. What else?
Yeah, they come in different types, mimicking the deep lung tissue, parenchyma, or the airways. Beyond infectious diseases, they're used for toxicology testing if inhaled substances are harmful and modeling complex conditions like pulmonary fibrosis, which involves scarring of the lung tissue. Their small size is a huge plus for high-throughput drug screening, too. Tests lots of compounds quickly.
Kidney organoids next. Given the wait list for kidney transplants, these seem crucial.
They really are. Kidney organoids can form structures that resemble nephrons, the kidney's filtering units, and collecting ducts. They're used to model genetic kidney diseases, assess whether drugs might be toxic to the kidney, nephrotoxicity, and they represent a really important step towards eventually bioengineering functional kidney tissue for transplantation.
And liver organoids. The liver's involved in so much, especially drug metabolism.
Absolutely essential. Liver organoids are key for modeling liver cancers, viral hepatitis like Hep B and C. They're significantly improving drug screening by better mimicking how human liver cells actually behave in vivo, especially how they metabolize drugs. And yes, like kidneys, there's huge interest in using them as building blocks for bioprinting transplantable liver tissue.
Okay, are there other really interesting ones emerging? Skeletal organoids, maybe?
Yes, skeletal organoids are a growing area. This includes models of bone, cartilage, even bone marrow, and the callus that forms during fracture healing.
So useful for osteoporosis,
Arthritis.
broken bones.
Exactly. Studying bone remodeling, diseases like osteoporosis, how fractures heal, and providing platforms for testing drugs or regenerative therapies for those conditions.
What about the heart? Cardiac organoids.
These life pumps are crucial too. They can beat spontaneously. They're vital for modeling cardiovascular diseases, straining drugs for potential cardiotoxicity heart damage, which is a major reason drugs fail late in development. They help analyze complex cellular interactions in conditions like heart attacks or heart failure.
It's amazing how diverse these are. Any other really notable ones?
Well, retinal organoids are fascinating. They model the light-sensitive tissue at the back of the eye. Really important because good animal models for human vision are lacking. Some have even shown intrinsic light sensitivity.
Wow.
There's also work on female reproductive organoids like trophoblast organoids mimicking the early placenta, crucial for understanding pregnancy. Inner ear organoids for hearing loss conditions like charge syndrome.
Male reproductive ones, too.
That area is still emerging, a bit less developed than others, but definitely important for understanding male infertility or cancers like prostate or testicular cancer. And I should mention nasal organoids. They can now be grown with cilia, those little hairs beating on their surface. That's great for studying colds, flu, how pathogens enter our airways.
Okay, this all sounds incredibly promising, almost revolutionary. But let's be realistic. What's the road ahead? What are the big challenges, the scientific headaches that researchers are still grappling with?
Right, it's not all smooth sailing. There are significant hurdles. One is incomplete maturation and lack of proper zonation,
Really?
meaning they often don't fully mature to an adult state, and they might not replicate the complex internal organization, the specific zones and cell distributions you find in a real organ. They're great models, but not perfect replicas yet.
And getting blood supply to them must be tough. Vascularization.
That's arguably the biggest challenge right now. Most organoids lack their own functional blood vessel network. This limits their size, their long-term survival, and their function because they can't get enough nutrients or remove waste effectively.
So how do you solve that?
People are trying different things. Co-culturing them with blood vessel organoids, trying to encourage the host's own blood vessels to grow into them after transplantation. It's a major focus of research. Using microfluidic chips helps too.
And if you do transplant them someday, immune rejection. Safety.
Absolutely critical concerns. How do you prevent the patient's immune system from attacking the organoid? Bioengineering approaches are key here, maybe encapsulating the organoids in special materials to hide them from the immune system.
Yeah.
And safety is paramount. You have to be absolutely sure there are no undifferentiated stem cells left that could potentially form tumors or unwanted tissues. Rigorous quality control is essential.
And just getting consistent results must be hard. Standardization.
Yes, that's a big one for the field. Right now, protocols can vary a lot between labs. This leads to variability in how well organoids grow, how mature they get. It hinders large-scale production and makes it harder to compare results globally. We need more standardized, robust methods.
So overcoming these hurdles,
Yeah.
what's it going to take? Where does the field need to go?
It's really a combination of things. We need even more advanced stem cell techniques, more sophisticated biomimetic materials, the synthetic hydrogels and scaffolds we talked about, getting even better. And just continuous optimization of the culture methods, the growth factors, the signaling molecules, really designing those precise tissue-specific microenvironments to nudge the organoids towards full maturity and function.
And I've heard artificial intelligence might play a big role here. Speed things up.
Oh, absolutely. AI is expected to be huge. Imagine using AI to analyze vast amounts of data from experiments, different cell types, media components, physical cues.
To find the perfect recipe faster.
Exactly. AI could help predict the optimal conditions much faster than traditional trial and error. It could really accelerate the process of overcoming these challenges and unlocking the technology's full potential.
Wow. Well, this deep dive has certainly shown how organoids are this incredible bridge, hasn't it?
Yeah.
Connecting that really cutting-edge lab science to potential real-world clinical impact.
They absolutely are, transforming how we understand disease, how we test drugs with more precision, maybe even how we eventually grow replacement
It's
tissues.
a genuine paradigm shift.
I really think so. It offers a future where our understanding of our own biology and how to fix it when it goes wrong is just much more intimate and precise.
So here's something to think about. If we can now grow these intricate miniature organs, mimicking human biology in such detail, what medical mysteries, things we thought were maybe insurmountable, might we finally unlock or even solve in the next few years?
Yeah.
The future of medicine. It
Yeah.
could literally be growing right now, right before our eyes.